Is my voice is okay?
Yes, it's okay.
Okay. Okay. So here we have a question. What is this signal of NF-κb in the cancer cell? I mean, this is the first question, but we have the second question. So what is this signal of the NF-κb in the cancer cells? Some of our participants ask about this question.
What is the signal?
Yeah, what is the signal of the NF-κb in the cancer cells? Is there any signal for it?
Okay. So I think they I think my laptop just froze. Okay. So I think they're talking about the signal. Okay, so about this signal,
We don't have your presentation. If you are just wanted to share it, or you are just explaining.
So is my presentation visible? Oh, it's not. Okay, okay. I think it's not. Yeah, this one. So do they mean by these signals?
I think one of our participants asked this question. Maybe you informed us about the signal. Maybe you'll explain the signal of NF-κb, any relation with the cancer cells, if you mentioned
Okay.
anything about.
So so actually the thing is that in cancer cells, the thing is that any of the signals which are received by the body in a normal functioning, cancer cells will use the same signaling. In normal, that's why it is very hard to track cancer whenever there is the misregulation of NF-κb. So since they both have the same signaling, which is being received by the receptors, they both we are not able to detect which signal is being transmitted by the cancers, or which NF-κb is problematic in that sense. Hence, we are not able to detect properly that. That's why we need to prepare the nanoparticles in such a way that they only target the cancer cells, because we are still not able to figure out that which signals are being transmitted by the cancer cells.
Okay, thank you. And we have the second question here. How successful the uptake of the nano antibodies in the cancer cell size range? I mean that's active or passive uptake in different sizes of the cancer cells. How is the uptake? Is it successful, and how you can explain this
for
issue?
now, okay. So the thing is, for now, is not very successful. It's not very successful. We are still, I think, in the mid of it. I would say there is like 30% success to that. But the hallmark one, which I just said about the silver citrate nanoparticles, they have worked very well in this case, because we have all the data that we need for the success. So for now, if I had just to consider the silver citrate nanoparticles, that are absolutely the best ones. But other methods which I discussed earlier, they are not that efficient. And many of the projects were done on the mice, but this one definitely shows a lot of promise. So I think for now I can say 30%, but in future it might be just above 70 to 80%.
Okay, I appreciate your answering the question, and thank you very much for your explanation, your project. Thank you.
Thank you for having me.